Today we are going to discuss about the hybridoma technology, which include the production and applications of monoclonal antibody, which is an important topic from pharmaceutical biotechnology of 6th semester BPharm Unit 3. So these are the some of the question which is seen in your university examination, which is usually seen as an 10-mark question as well as 5-mark question as well as 2 marks. So this is a repeated question also. Define hybridoma technology. Explain the production and application of the monoclonal antibodies. And same question is seen as an 5-mark also. So you have to write the definition and the steps. So based on this, this is the learning objective of the today's session. By the end of the session, students will be able to understand, describe and write about the production, application and definition of production of monoclonal antibody, which is done by a principle of hybridoma technology. Okay. So let's start with the definition. What is mean by hybridoma technology? So as the word indicate, it is an hybrid. It is a formation of hybrid by producing or by fusing antibody producing cells like beta lymphocyte cells with some of the immortalized cell lines like myeloma cells. So what is the speciality of hybridoma technology? So as we all know, the beta lymphocyte cells are the responsible cells which is required for the production of antibody in an immune system. So the problem with the beta lymphocyte cells are which have a limited lifespan, so it will be destroyed very soon. So by fusing this beta lymphocyte cells which have a capacity to produce antibody with some immortalized cells, that means which will not be dead, which have a long life period like myeloma cells. ഇതാണ് ഇതിൻ്റെ ബേസിക് ആയിട്ടുള്ള പ്രിൻസിപ്പിൾ എന്ന് പറയുന്നത്. ഒരു ഹൈബ്രിഡ് ആയിട്ടുള്ള സെല്ല് ഫോം ചെയ്യുക എന്നുള്ളതാണ് ഹൈബ്രിഡോമ ടെക്നോളജി എന്ന് പറയുന്നത്. എങ്ങനെയുള്ള ഹൈബ്രിഡ് സെൽസുകളാണ് ഫോം ചെയ്യുന്നത്? By fusing the antibody producing cells like beta lymphocyte cell with some myeloma cell which are immortalized cell lines which do not have any kind of death ഇല്ലാത്ത ടൈപ്പ് സെൽസുകൾ യൂസ് ചെയ്തിട്ട് ആൻറ്റിബോഡി പ്രൊഡ്യൂസ് ചെയ്യാൻ എബിലിറ്റി ഉള്ള ബീറ്റാ ലിംഫോസൈറ്റ് സെല്ലുകളുമായിട്ട് ഫ്യൂസ് ചെയ്തിട്ട് അൺലിമിറ്റഡ് ആയിട്ടുള്ള ലൈഫുള്ള ആൻറ്റിബോഡി പ്രൊഡ്യൂസ് ചെയ്യുന്ന ബീറ്റാ ലിംഫോസൈറ്റ്സുകൾ ഉണ്ടാക്കുന്ന ടെക്നോളജിനെയാണ് ഹൈബ്രിഡോമ ടെക്നോളജീസ് എന്ന് പറയുന്നത്. ഓക്കെ, now what is mean by monoclonal antibodies? So these are the single antibodies which are made by fusing antibody producing cells with immortalized cell lines which will result in a cell called hybridoma. അങ്ങനെ ഉണ്ടായിട്ടുള്ള സെല്ലിനെ അല്ലെങ്കിൽ ആൻറ്റിബോഡിനെയാണ് നമ്മൾ മോണോക്ലോണൽ ആൻറ്റിബോഡി എന്ന് പറയുന്നത്. And which was discovered by two scientists, George Kohler as well as Cesar Milstein. So these are the scientists who discovered this hybridoma technology. So what is the principle of hybridoma technology? എന്താണ് hybridoma technologyയുടെ principle എന്ന് പറയുന്നത്? So as I mentioned, we are going to prepare an cell which have an ability of immortality and also antibody producing capacity. So in order to produce this immortal antibody production cells, we have to take one cell which have an ability to produce antibody production, that is beta lymphocyte cell, and we have to take another cell that is myeloma cell which have an capacity of immortality. ഓക്കെ, അപ്പോൾ അൺലിമിറ്റഡ് ആയിട്ടുള്ള ലൈഫ് സ്പാൻ ഉള്ള ഒരു സെല്ലും ആൻറ്റിബോഡി പ്രൊഡ്യൂസ് ചെയ്യാനുള്ള ഒരു സെല്ലും എടുത്തിട്ട് അതിനെ ഫ്യൂസ് ചെയ്യിക്കുക എന്നുള്ള ടെക്നോളജിനെയാണ് നമ്മൾ ഹൈബ്രിഡോമ ടെക്നോളജി എന്ന് പറയുന്നത്. സോ ആസ് ഐ മെൻഷൻ, ദ ലിമിറ്റേഷൻ ഓഫ് ബീറ്റാ ലിംഫോസൈറ്റ് സെൽസ് ഈസ് വിച്ച് ഹാവ് an ability to produce antibody but which have an limited lifespan. Okay, but the myeloma cells which do not have any capacity to produce antibody but they have an immortality. So so which have a long lifespan. So we will compare this two property, sorry, we will combine this two property to produce an hybrid cell which have an ability of immortality as well as antibody production. So that is the hybridoma technology. This is the basic principle of hybridoma technology. Now, before starting what is the production of monoclonal antibody, just we have to understand what is the difference between monoclonal antibody and polyclonal antibody. So as the name indicates, monoclonal antibodies are that identical cells or antibody that are identical because they are produced by one type of immune cells, that is beta lymphocyte cells, of a single parent which will bind to a single specific antigen. So ഏത് ആൻറ്റിജൻ ആണോ ആൻറ്റിബോഡി ഉണ്ടാക്കാൻ റെസ്പോൺസിബിൾ ആയിട്ടുള്ള ബീറ്റാ ലിംഫോസൈറ്റ് സെൽ, ഓക്കെ, ആ ആൻറ്റിജനെതിരെ ആക്ട് ചെയ്യുന്ന ആൻറ്റിബോഡി മാത്രം ഒരു സിംഗിൾ പാരന്റ് ബീറ്റാ ലിംഫോസൈറ്റ് സെല്ലിൽ നിന്ന് പ്രൊഡ്യൂസ് ചെയ്യുന്ന ആൻറ്റിബോഡിനെയാണ് മോണോക്ലോണൽ ആൻറ്റിബോഡീസ് എന്ന് പറയുന്നത്. ഓക്കെ, and the antibody which is produced from the multiple clones of beta lymphocyte cells, therefore they bind to the number of different epitopes of the antigen like IV immunoglobulin, so which is not specific against and specific antigen. Polyclonal antibodies are the antibody which will act against different kinds of antigen. Monoclonal antibody will act against a single specific antigen. And regarding the production process is expensive for the expensive process in case of monoclonal antibody, and it is inexpensive in case of polyclonal antibody, because this is as simple as that of production of serum. Antibody containing preparation എങ്ങനെയാണോ ഉണ്ടാക്കുന്നത് അത്രയും പണിയേ ഉള്ളൂ polyclonal antibody ഉണ്ടാക്കാൻ. But monoclonal antibody ഉണ്ടാക്കാൻ there are many steps, okay, and which required a long production time for the production of monoclonal antibody, and polyclonal antibody production is very rapid, and large quantity of specific antibody can be produced by monoclonal antibody. Here we will get the large quantity of non-specific antibody, and monoclonal antibody will recognize a single epitope of an antigen, and polyclonal antibody will recognize multiple epitopes of an antigen, and production is continuous and uniform in case of monoclonal antibody production. And polyclonal antibody, there is a batch difference variation in composition while production, because each time we will get new antibodies. But in case of monoclonal antibody, if we are producing the monoclonal antibody, it will be continuous and it will be uniform. Okay, so that is the difference between monoclonal antibody and polyclonal antibody. So what is the advantage of monoclonal antibody over the polyclonal antibody? So these are the some of the advantages. This is very specific, they have very specific target, because this monoclonal antibody will bind with the only those antigen which is very specific. So this is the latest technology which is mainly used for the treatment of many cancers. There was a news in the newspaper, we had discovered a drug against some kind of cancer. So that was a monoclonal antibody, because the cancer which is caused by some specific antigen is treated by the antibody which is produced very specifically against the same antigen. So that it will be very expensive. We have to identify the specific antigen and we have to prepare antibody at respective time, and do not have any intracellular activity, so it will not produce any kind of side effect as well as reaction, because it will only react with the specific antigen, and it is not metabolized by the kidney or liver, but it is catabolized within the cell, resulting in amino acid which is recycled within the body. So these are the advantages of monoclonal antibodies. Okay, now coming to the steps. So this is the very important part of the session. If there is a question regarding monoclonal antibody production or hybridoma technology, you must enlist the steps of monoclonal antibody production or hybridoma technology. So these are the different steps. There are 10 different steps for hybridoma technology. So first one is immunization. Second one is screening of mice for antibody production. Third one is preparation of myeloma cells. And fourth one is fusion of myeloma cells with immune spleen cells. And fifth one, selection of hybridomas. Sixth one, screening the product. Seven, cloning and propagation. Eighth, production of monoclonal antibody by fermentation process. Then purification of monoclonal antibodies, and finally characterization and storage. So these are the different steps. So you must write the different steps, whether the question is for 2 mark or 5 mark or 10 mark. You must write the steps first, and based on the mark, if it is for 5 marks or if it is for 10 marks, you have to elaborate each steps of this hybridoma technology in detail. So now we will discuss one by one. First one, so first step is immunization. So we have already mentioned we are going to produce an antibody which is very specific against an antigen. So in order to produce a monoclonal antibody against the specific antigen, first we have to identify the antigen, and that antigen has to be injected into an animal like mice or mouse to immunize it, to produce antibody inside the animals. Okay, so whether which antigen has to be, which antibody has to be prepared, the specific antigen should be injected into the mice or mouse. അപ്പോൾ നമുക്ക് ഏത് ആൻറ്റിബോഡിയാണോ വേണ്ടത്, ആ ആൻറ്റിബോഡി ഉണ്ടാക്കാൻ റെസ്പോൺസിബിൾ ആയിട്ടുള്ള ആൻറ്റിജൻ ഏതാണോ ആ ആൻറ്റിജനാണ് നമ്മൾ അനിമൽസ് ലൈക്ക് മൈസ് ഓർ മൗസിലോട്ട് ഇഞ്ചെക്ട് ചെയ്യുന്നത്. ഓക്കെ, സോ നമ്മൾ സാധാരണഗതിയിൽ അതിനെ ഇഞ്ചെക്ട് ചെയ്യുന്നത് നമ്മൾ അതിൻ്റെ ടൈലുള്ള വെയിനിൽ വഴിയാണ്, ടൈല് അതിൻ്റെ വാലിലുള്ള വെയിൻ വഴിയാണ് നമ്മൾ ഈ പറയുന്ന സാധനം ഇഞ്ചെക്ട് ചെയ്യാറുള്ളത്. So first we will immunize the mice or mouse with appropriate antigen for every 2 to 3 weeks. So നമ്മൾ ഒരു പ്രാവശ്യം ഇഞ്ചെക്ട് ചെയ്ത് 2 to 3 week വെയിറ്റ് ചെയ്യും. Then we will screen for the second step, that is whether there is insufficient production of antibody or not. നമ്മളത് സ്ക്രീൻ ചെയ്യും, and if it is low level of antibody, again after the 2 to 3 week we will again inject the same antigen. Okay, so usually this antigen is prepared for injection either by emulsifying with antigen with foreign adjuvants or other adjuvants by homogenizing gel slices that containing antibodies. So this is the steps. First one is immunization, antigen with some kind of adjuvants. We will emulsify it and we will inject into the antigen. So what will happen? We are giving an antigen, which means we are artificially immunizing, we are artificially producing antibody in the animals. അല്ലേ, നമ്മൾ ആൻറ്റിജൻ കൊടുക്കുന്ന സമയത്ത് സ്വാഭാവികമായിട്ട് എന്ത് സംഭവിക്കും, ആ അനിമലിലുള്ള ഇമ്മ്യൂൺ സിസ്റ്റം ആക്ടിവേറ്റഡ് ആയിരിക്കും. ആക്ടിവേറ്റ് ആവുന്ന സമയത്ത് അതിൻ്റെ ഇമ്മ്യൂൺ റെസ്പോൺസസുകൾ ഉണ്ടാവും. ഇറ്റ് വിൽ പ്രൊഡ്യൂസ് ആൻറ്റിബോഡി അഗൈൻസ്റ്റ് ദ ആൻറ്റിജൻ. ഓക്കെ, now coming to the second step, that is the screening of mice for antibody production. So once the antigen is injected into the animals in every 2 to 3 weeks, we will draw the blood sample from the mice for the measurement of serum antibody by different technology like ELISA, ഫ്ലോ സൈറ്റോമീറ്റർ, ഇങ്ങനെയുള്ള മെത്തേഡ് യൂസ് ചെയ്യുന്നതാണ്. നമ്മൾ ആൻറ്റിബോഡി എത്രത്തോളം ഉണ്ടായിട്ടുണ്ടെന്ന് മെഷർ ചെയ്യാൻ വേണ്ടിയിട്ടാണ് ഈ ഒരു മെത്തേഡ്സുകൾ നമ്മൾ യൂസ് ചെയ്യുന്നത്. ഓക്കെ, എലൈസ, ഫ്ലോ സൈറ്റോമീറ്റർ ഒക്കെയാണ് നമ്മൾ അതിന് വേണ്ടിയിട്ട് യൂസ് ചെയ്യുക. 2 to 3 വീക്ക് കഴിഞ്ഞിട്ട് നമ്മൾ കൊടുത്ത ആൻറ്റിജന് സ്പെസിഫിക് ആയിട്ടുള്ള ആൻറ്റിബോഡി എത്രത്തോളം മൈസിൽ ഉണ്ടായിട്ടുണ്ട് എന്ന് ഐഡൻറ്റിഫൈ ചെയ്യാൻ വേണ്ടി ബ്ലഡ് എടുക്കും, നമ്മൾ സാമ്പിൾ എടുക്കും. ഓക്കെ, നൗ ആഫ്റ്റർ മോണിറ്ററിങ് ദ സിറം ആൻറ്റിബോഡി, ബ്ലഡിലുള്ള ആൻറ്റിബോഡി മെഷർ ചെയ്യുന്ന സമയത്ത് ഇഫ് ദ ടൈറ്റർ ഈസ് ടു ലോ, അപ്പോൾ അതിൽ ആൻറ്റിബോഡി പ്രൊഡക്ഷൻ കുറവാണെന്നുണ്ടെങ്കിൽ we will boosted with antigen until adequate response is achieved in the animals. If antibody production is high enough, we will finish the process by injecting without the adjuvant intraperitoneally or by intravenously through the tail vein. Okay, so usually we will do this 3 days before the fusion. So there will be an fusion of മൈലോമ സെൽ ആസ് വെൽ ആസ് ബീറ്റാ ലിംഫോസൈറ്റ് സെൽസ്. സോ ആ ഒരു പ്രോസസ്സ് നമ്മൾ പ്ലാൻ ചെയ്യുന്നതിന് 3 days ബിഫോർ നമ്മൾ എന്ത് ചെയ്യും, വി വിൽ ഗീവ് ആൻ ബൂസ്റ്റർ ഡോസ് ആൻഡ് 2 weeks ആഫ്റ്റർ ദ പ്രീവിയസ് ഇമ്മ്യൂണൈസേഷൻ. അപ്പോൾ 2 to 3 weeks ഫ്രീക്വൻ്റ് ആയിട്ടാണ് നമ്മൾ ഇമ്മ്യൂണൈസേഷൻ ചെയ്യാൻ നമ്മൾ ഫസ്റ്റ് സ്റ്റെപ്പിൽ പറഞ്ഞു. 2 weeks കഴിഞ്ഞതിന് ശേഷം നമ്മൾ ഫ്യൂസ് ചെയ്യുന്ന ആ ഒരു പ്രോസസ്സ് എടുക്കാൻ ഉദ്ദേശിക്കുന്നതിന് 3 days മുന്നേ വീണ്ടും നമ്മളൊരു ബൂസ്റ്റർ ഡോസ് കൊടുത്തിട്ട് നമ്മൾ ഇമ്മ്യൂണൈസേഷൻ എന്ന് പറയുന്ന പ്രോസസ്സ് സ്റ്റോപ്പ് ചെയ്യും. ഓക്കെ, സോ ആഫ്റ്റർ അച്ചീവിങ് സഫിഷ്യൻറ്റ് ആൻറ്റിബോഡി ടൈറ്റർ, the immunized mice will be euthanized. We will sacrifice the mice and then we will remove the spleen where the beta lymphocytes will be there, and this spleen cell will be used as an beta lymphocyte cells source for the fusion of myeloma cells. So that is the second step, screening of mice for antibody production. So we will take the spleen of the immunized mice after euthanizing it. So while writing answer, you have to make sure that you have to write the terms like euthanization or sacrification. You should not use any words like by killing the animals. So as per the എത്തിക്സ് അല്ലെങ്കിൽ നമ്മൾ എന്താ പറയുക, ക്രൂവാലിറ്റി ഒരു പർപ്പസിന് വേണ്ടിയിട്ടാണ് അനിമൽസിനെ നമ്മൾ ഇത് ചെയ്യുന്നത്. So we never ever use the term kill in case of animal studies. We have to use either euthanization or we have to use either sacrification of the animal. Okay, നമ്മൾ അങ്ങനെ ടേം യൂസ് ചെയ്യാൻ പാടുള്ളൂ. സോ വൈൽ റൈറ്റിംഗ് ദ ആൻസർ യു മസ്റ്റ് റൈറ്റ് either euthanizing or by sacrifice. Do not use kill. Okay, so after euthanizing the animal, we will remove the spleen and we will take it as a source for beta lymphocytes for fusion with the myeloma cell. So now we have the cells which is responsible for the antibody production, that is the beta lymphocyte cell, that is the spleen of the immunized mice. Now we have to prepare the myeloma cell which have an ability to immortality, ലൈഫ് ടൈം ആയിട്ടുള്ള ഒരു എബിലിറ്റി ഉള്ള ലൈഫ് ലോങ് എബിലിറ്റി ഉള്ള ഒരു പ്രോപ്പർട്ടി ഉള്ള സെല്ലാണ് മൈലോമ സെൽസ് എന്ന് പറയുന്നത്. സോ usually myeloma cells are immortal. So in order to produce the myeloma cell which is ready for the fusion with the beta lymphocyte cell, we have to treat or we have to culture the myeloma cell with 8-azoguanine. So this is the chemical which is used to make sure to ensure their sensitivity towards hypoxanthine, aminoterine, thymidine medium. So I will explain what is this, the role of hypoxanthine, aminoterine, thymidine in the next slide. So this is the reagent which is used to ensure the sensitivity of myeloma cell towards the HAT medium, that is hypoxanthine, aminoterine, thymidine medium. And the peculiarity of this myeloma cell which have an high viability and rapid growth. So these are the some of the examples of the myeloma cell: sp2-0, ag14, NSO, p3x633 ഇങ്ങനെയുള്ള പേരുകളൊക്കെയാണ് ഇതിന് എക്സാമ്പിൾ ആയിട്ടുള്ളത് മൈലോമ സെൽസിനുള്ള എക്സാമ്പിൾ. നൗ coming to the fourth step, that is the fusion of myeloma cell with the immune spleen cells. So this is the very important step, and this is a very crucial step of the hybridoma technology, because after thorough washing of the lymphocytes which is taken from the spleen of the animal, we have to treat the lymphocyte cells with the 8-azoguanine treated myeloma cell with polyethylene glycol. PEG ആണ് ഇവിടെ നമ്മൾ യൂസ് ചെയ്യുന്ന reagent in order to fusion. Okay, so for a few period we will use this HGPRT defective myeloma cells with the lymphocytes which have an HGPRT cells. So with the polyethylene glycol. Okay, അപ്പോൾ ഫ്യൂഷന് വേണ്ടിയിട്ട് യൂസ് ചെയ്യുന്ന റിയേജൻറ്റ് ഏതാണെന്ന് ചോദിച്ചു കഴിഞ്ഞാൽ പോളിയെത്തിലിൻ ഗ്ലൈക്കോൾ ആണ് അവിടെ യൂസ് ചെയ്യുന്ന റിയേജൻറ്റ്. ആൻഡ് ആഫ്റ്റർ ദ ഫ്യൂഷൻ PEG is removed by washing and cells are kept in the fresh medium, and these cells are composed of hybridoma cells, free myeloma cells as well as free lymphocyte cells. അതായത് ആഫ്റ്റർ ദ ഫ്യൂഷൻ we will get 3 type of cells. One, hybridoma cells, in which the beta lymphocytes will fused with the myeloma cells, that is the hybridoma cells. Then there will be some free myeloma cell which we had taken for the treatment, and we have some free lymphocyte cell which do not fuse each other. Okay, so fusing antibody producing spleen cell with the cell derived from the immortal tumor lymphocyte cell will result from hybridoma that have an ability of unlimited growth with antibody production. Okay, അപ്പോൾ ഈ രണ്ട് സെല്ലിൻ്റെയും റോള് നിങ്ങൾ മനസ്സിലാക്കി വെക്കുക. In case of spleen cells which have an ability to produce antibody but it have an limited lifespan, and the role of immortal cell which have an ability to produce which have an ability of immortality but do not have any ability to produce antibody. So we are going to combine these two property of immortality of myeloma cell with the antibody production capacity of the beta lymphocytes to produce an cell which have an immortal antibody producing capacity. Okay, so that is the third step. Now coming to the next step. So this is the steps till now we had explained. So first we will inject the antigen into the animal like mice. So we will get some kind of antibody in the spleen, and we will use the myeloma cells which is lack of HGPRT. So we will fuse this together with the polyethylene glycol to produce some kind of myeloma cells, unfused myeloma cell, unfused beta lymphocyte cell, along with the fused myeloma cells, okay, hybrid cells. Now next step is selection of hybridoma cell. So we don't know, it is not as simple as that every cell will fuse with the myeloma cell as well as beta lymphocyte cells. We cannot predict that. So we have to select the hybrid cell which is fused with the myeloma cell as well as beta lymphocytes cell from the unfused myeloma cell as well as unfused lymphocyte cells. Okay, അപ്പോൾ നമുക്കിപ്പോൾ ഫ്യൂസ് ചെയ്ത സമയത്ത് നമുക്ക് 3 ടൈപ്പ് സെൽസുകളാണ് കിട്ടിയെന്ന് പറഞ്ഞു. ഏതൊക്കെയാണ് 3 ടൈപ്പ് സെൽസ്? ഒന്ന് ഹൈബ്രിഡോമ സെൽസ് ആണ്, രണ്ടാമത്തത് ഫ്രീ ആയിട്ട് നിൽക്കുന്ന മൈലോമ സെൽസുകളാണ്, ഫ്രീ ആയിട്ട് നിൽക്കുന്ന ബീറ്റ ലിംഫോസൈറ്റ് സെൽസുകളാണ്. ഇതിൽ നിന്ന് ഹൈബ്രിഡ് സെൽസിനെയാണ് നമുക്ക് സെപ്പറേറ്റ് ചെയ്യേണ്ടത്. സോ in order to produce this, in order to select the hybridoma cell, we will use an principle of the de novo synthesis process. So I think you have already aware about this process, de novo synthesis and process which is required for the production of DNA. So the production of replication of DNA. So replication of DNA is required for the further replication of any cell. So even it is hybrid cell also, it requires some kind of de novo process. So usually de novo process occur it will start from dihydrofolate by using it will be converted to tetrahydrofolate, and it will be converted to nucleotide and DNA. And another method for synthesis of nucleotide is this salvage pathway, which will use hypoxanthine to produce nucleotide by using HGPRT, which is an enzyme responsible for the hypoxanthine guanine, which is an enzyme hypoxanthine guanine phosphoribose transferase, which is responsible for the conversion of hypoxanthine to nucleotide. Okay. Then there is another pathway, that is thymidine is converted to nucleotide by using thymidine kinase. So what is the role of this pathway in the hybridoma technology? We have mentioned 2 types of cell. One is beta lymphocyte cells as well as myeloma cells. In this, myeloma cells do not have any kind of HGPRT. Okay. മൈലോമ സെല്ലിന് സ്വന്തമായിട്ട് HGPRTase എന്ന് പറയുന്ന എൻസൈം ഇല്ല. ഓക്കെ, HGPRTase എൻസൈം ഇല്ല. അപ്പോൾ മൈലോമ സെല്ല് സ്വാഭാവികമായിട്ടും എന്തായിരിക്കും സംഭവിക്കുന്നുണ്ടാവുക? ഇഫ് യു ആർ ഗ്രോയിങ് ഇൻ ആൻ മീഡിയം, ഹാറ്റ് മീഡിയം, ഹൈപ്പോസാന്തിൻ അമിനോട്ടറിൻ തൈമിഡിൻ മീഡിയത്തിൽ നമ്മൾ ഗ്രോ ചെയ്ത് കഴിഞ്ഞ് കഴിഞ്ഞാൽ വാട്ട് വിൽ ഹാപ്പൻ? Since it do not have any kind of HGPRTase, it cannot produce the nucleotide, and so it cannot replicate itself to produce new progenies of the myeloma cells. Okay, even though they are immortal, they do not have any HGPRTase enzyme, so it cannot replicate itself. Okay, but the the property of beta lymphocyte cell, they have an ability to produce antibody, and they have an ability, they have an enzyme, HGPRT enzyme, so it can replicate itself, but they have like limited lifespan. They have limited lifespan. Okay, limited lifespan കഴിയുന്ന സമയത്ത് അത് ഓട്ടോമാറ്റിക്കായിട്ട് ഡെഡ് ആവും. Okay, so by fusing these two cells, one cell have an HGPRT enzyme and myeloma cell do not have an HGPRT cell, but the beta lymphocyte cell have an HGPRT as well as antibody producing capacity, and myeloma cell have only immortality. So by fusing these two cells, we are fusing the gene which is responsible for this HGPRT enzyme. So the immortality cells like myeloma cell will get this HGPRT. So only fused cell will grow in HAT media. എന്തുകൊണ്ടാണ് അങ്ങനെ പറയാൻ കാരണം? ഹാറ്റ് മീഡിയത്തിൽ ഫ്യൂസ്ഡ് സെല്ലുകൾ മാത്രം ഗ്രോ ചെയ്യുള്ളൂ എന്ന് പറയാൻ കാരണം എന്താണ്? ഫ്യൂസ് ചെയ്ത സെല്ലിന് മാത്രമാണ് HGPRT എന്ന് പറയുന്ന എൻസൈം ഉണ്ടാവുള്ളൂ. ഓക്കെ, അതിന് മാത്രമേ ആ മീഡിയത്തിൽ റെപ്ലിക്കേറ്റ് ചെയ്യാൻ പറ്റുള്ളൂ. ക്ലിയർ ആണെന്ന് വിചാരിക്കുന്നു. HGPRT എന്ന് പറയുന്ന എൻസൈം ഉണ്ടെങ്കിൽ മാത്രമേ ഏത് മീഡിയത്തിൽ ന്യൂക്ലിയോടൈഡ് ഉണ്ടാക്കാൻ പറ്റുള്ളൂ? ഹൈപ്പോസാന്തിൻ, തൈമിഡിൻ, അമിനോടെറിൻ എന്ന് പറയുന്ന മീഡിയത്തിൽ ന്യൂക്ലിയോടൈഡ് ഉണ്ടാക്കണമെന്നുണ്ടെങ്കിൽ എന്ത് വേണം? HGPRT എൻസൈം വേണം. ഓക്കെ, അപ്പോൾ ഇവിടെ ആൻറ്റിബോഡി പ്രൊഡ്യൂസിങ് സെൽസുകളായിട്ടുള്ള ബീറ്റാ ലിംഫോസൈറ്റ്സിന് HGPRT ഉണ്ട്, ആൻറ്റിബോഡി പ്രൊഡ്യൂസിങ് എബിലിറ്റി ഉണ്ട്, ബട്ട് ദേ ഹാവ് ലിമിറ്റഡ് ലൈഫ് സ്പാൻ. മൈലോമ സെല്ലിലോട്ട് വരുന്ന സമയത്ത് ദേ ഹാവ് ഇമ്മോർട്ടാലിറ്റി, ബട്ട് ദേ ഡു നോട്ട് ഹാവ് HGPRT enzyme. So if we are growing the mixture of these three cells that we will get after the fusion, ഏതൊക്കെയാണ് 3 cells? Unfused beta lymphocyte cells, unfused myeloma cells and fused hybridoma cells. ഇതാണ് 3 type cells. Since the unfused beta lymphocytes have a limited lifespan, so it will die after the lifespan. 12-24 days കഴിഞ്ഞാൽ അത് ഡെഡ് ആവും. And unfused myeloma cell, they do not have HGPRT enzyme, so they cannot replicate in the HAT medium, so they will also die. The only cells which is fused, which have an HGPRT enzyme, which have an antibody producing ability and which have an ability of immortality will replicate only in the HAT medium. So that is the selection process of hybridoma cells. So due to the presence of HGPRT enzyme, the cell which are deficient in the HGPRT, that is the unfused mutated myeloma cells, will cannot survive due to inhibition of de novo synthesis of purine nucleotide. Okay, അപ്പോൾ അൺഫ്യൂസ്ഡ് ആയിട്ടുള്ള മൈലോമ സെല്ലിൻ്റെ പ്രശ്നം എന്താണ്? ദേ ഡു നോട്ട് ഹാവ് HGPRT enzyme. ഹൈപ്പോസാന്തിൻ, ഗുവാനിൻ, ഫോസ്ഫോറൈബോസിലേസ്, ട്രാൻസ്ഫറേസ് എന്ന് പറയുന്ന എൻസൈം ഇല്ലാത്ത സെൽസുകളാണ് ഇത് അൺഫ്യൂസ്ഡ് ആയിട്ടുള്ള മൈലോമ സെൽസുകൾ. സോ they cannot survive in the HAT medium. What is HAT medium? Hypoxanthine, aminoterin, thymidine medium. That is the HAT medium. So they cannot survive in the HAT medium due to inhibition of de novo synthesis because they do not have this HGPRT enzyme. Okay, they do not have the HGPRT enzyme, so they cannot synthesize the nucleotide, so they cannot replicate also. Okay, so that is the property of hybridoma, myeloma cells. Now what is the limitation of unfused normal spleen cell? They cannot grow in the HAT medium because they have a limited lifespan. Okay, after the lifespan they will dead. Now only the hybridoma cell which have an ability of myeloma cell to grow in vitro HAT medium and have an functional HGPRT gene which is obtained from the lymphocytes will grow and proliferate in the heart medium. So that is the fifth step, selection of the hybridoma cells. So only the hybridoma cells will grow in the hat medium, and this will happen usually from 7 to 10 days. Usually it will happen in 7 to 10 days. Okay. Now, so this is the selection process of hybridoma technology. Now coming to the next step, that is screening of the product. So we are getting some kind of hybridoma cell, right? So we will periodically test it for desired antibody specificity. We are getting an hybridoma cell which have an ability to produce antibody against a specific antigen. So in order to identify that the specificity of the antibody, we will use some technology like ELISA, radioimmunoassay, which we will discuss in later in the biotechnology classes. Okay, അപ്പോൾ അത് നമ്മൾ കുറച്ച് കഴിഞ്ഞ് നമ്മളത് ഡിസ്കസ് ചെയ്യുന്നതാണ്. ആ സമയത്ത് നമുക്കത് കൂടുതലായിട്ട് എക്സ്പ്ലെയിൻ ചെയ്യാം. ഓക്കെ, both as a antibody will bind with the specific antigen and unbounded antibody, other compound medium will be washed off so that we will identify the antibody which is referred as a monoclonal antibody. ഏത് ആൻറ്റിജനെതിരെയാണോ നമ്മൾ ഉണ്ടാക്കിയിട്ടുള്ള ആൻറ്റിബോഡി അതിനെതിരെ മാത്രമേ ഈ പറയുന്ന ആൻറ്റിബോഡി നമുക്ക് കിട്ടുള്ളൂ. ഓക്കെ, now coming to the seventh step, that is cloning and propagation. So we are getting some kind of hybridoma cell, right, which have an ability to produce antibody and which have an ability of immortality also. Now we have to increase the number of the hybridoma cell. So single hybrid cell producing desirable antibody are isolated and cloned in order to increase the number of hybridoma cells. So there are two methods for producing monoclonal antibody, that is the limiting dilution method as well as soft agar method. These are the two methods which is mainly used for cloning and propagation. So in case of limiting dilution method, the suspension of hybridoma cell is seriously diluted and allocates of each dilution are put into the microculture tube, and these dilutions are made that each liquids in the allocates in the well containing only single hybrid cell, and this ensure the antibody produced is monoclonal antibody. And in case of soft agar method, hybridoma cells are cultured in the soft agar. So one is in liquid media, one is in solid media, semi-solid ആയിട്ടുള്ള ഒരു മീഡിയത്തിലാണ് നമ്മൾ കൾച്ചർ ചെയ്യുന്നത്. ഓക്കെ, so in actual practice both the above technique are combined and used for the maximal production of the monoclonal antibody. Now coming to the production of monoclonal antibody. So ഇവിടെ നമ്മൾ എന്താ ചെയ്തത്, ക്ലോണിങ് ആൻഡ് പ്രൊപ്പഗേഷനിൽ we are increasing the number of hybridoma cells. Hybridoma cellന്റെ പ്രത്യേകത എന്താണ്? They have an ability to produce antibody and also they are immortal. Okay, so now we have to produce the monoclonal antibody. So in order to produce the monoclonal antibody, we have to culture the hybridoma technology, culture hybrid cells. So there are two methods for production of monoclonal antibody. One is in vitro tissue culture method and second one is in vivo animal method. So as we mentioned, in vitro tissue culture method, we will do this in an external environmental by giving some kind of simulated condition of the cells. So the simplest approach for producing monoclonal antibodies in vitro are to grow the hybridoma cell in a culture media. Usually for the culturing of the hybridoma cells, we will use fetal bovine serum which is used in tissue culture media containing bovine immunoglobulin, which can account for the substantial fraction of the immunoglobulin present in the culture fluid. But there are some disadvantages for this in vitro tissue culture techniques. That is the production process, the availability of monoclonal antibody will be low. Production of monoclonal antibody will be low in the culture bottle. So in order to improve this disadvantage, we will go for the second method, that is the in vivo animal method. So So this is usually done in hydrate cells as well as ascites in the peritoneal cavity of the mice. So usually this ascites means it's a pathological accumulation of a fluid in the peritoneal cavity of the mice. So ആ ഒരു പെരിറ്റോണിയൽ ക്യാവിറ്റിയിലാണ് നമ്മൾ ഈ മോണോക്ലോണൽ ആൻറ്റിബോഡിയുടെ കൾച്ചർ യൂഷ്വലി ചെയ്യുന്നത്. യൂഷ്വലി കണ്ടെയ്ൻ 5 to 20 milligram of monoclonal antibody per ml of this ascites fluid. Okay, so this is far superior when compared with the in vitro method, but this also have some kind of disadvantages. So collection is very risky. Animal kill ചെയ്യാതൊന്നും എടുക്കാൻ പറ്റില്ല, and there are some chances of contaminations or pathogens of microorganisms, and we have to sacrifice many animals for culturing the or production of monoclonal antibody. That is another disadvantages of in vivo animal method. So that is the 9th step, production of monoclonal antibody. And next step is purification of monoclonal antibody. So we will get an monoclonal group of monoclonal antibody. So this step is mainly done to produce reliable and predictable product which is suitable for the use in the human. This is done by removing the impurities such as host cell protein, DNA, advantageous as well as endogenous viruses, endotoxins and aggregates. So these are the some of the impurities which is usually present in the monoclonal antibody fluids. So we will remove this kind of impurities, and we will go for some kind of purification process like chromatographic techniques പോലെയുള്ള സാധനങ്ങളിലോട്ടൊക്കെയാണ് പോവുക. So this is the steps for purification of monoclonal antibody. So after the fermentation process, after the animal cell culture, we will harvest and we will centrifuge and we will filter it to remove the cell debris prior to the chromatography. Then we will go for protein A chromatography so that we will yield an highly purified product in a single step. Then we will go for low pH hold for viral inactivation. So നമ്മൾ അതിൻ്റെ pH acidic pHലോട്ട് കൊണ്ടുവന്നിട്ട് we will inactivate the endogenous and advantageous viruses. And additionally we will go for polishing chromatography for removal of product and process related impurities and viruses. Then we will go for an additional polishing chromatography for removal of product and process related impurity of viruses. Then viral filtration to remove endogenous and advantageous virus, and finally ultra filtration or diafiltration to formulate the bulk drug substance. So this is the purification of monoclonal antibody. And finally the characterization and storage. So here we will go for biochemical and physiochemical characterization for desired specificity of the monoclonal antibody for its stability, etc. And finally we will store in a liquid nitrogen at -196 degrees Celsius at several stage of cloning as well as culture. So this method is known as cryopreservation. Liquid nitrogen le nammal preserve cheyuna method neയാണ് നമ്മൾ we used to call it as an cryopreservation method. Cryopreservation. So usually monoclonal antibodies are stored in cryopreservation. Okay, so this is the steps of hybridoma technology. So we will summarize the steps of hybridoma technology. First we will immunize the mice with an antigen so that it will get immunized and it will produce antibody inside the animal, and which have an ability, beta lymphocytes cell have an ability of producing antibodies as well as it have an HGPRTase enzyme. And we will produce myeloma cell by treating with 8-azaguanine, which lack of HGPRTase, which lack of antibody production ability, which have an immortality, and we will fuse these two cells together with polyethylene glycol, so that we will get three type of cells: unfused myeloma cells as well as unfused beta lymphocyte cell. So from this fused beta lymphocyte cells with the myeloma cells, we will select the hybrid cell by culturing the cells in an HAT medium. So only the hybrid cells which is fused with the myeloma cells as well as beta lymphocytes will grow in the HAT medium, so that we can isolate that and we can clone it. We can propagate the number of hybrid cells. Then we will produce the monoclonal antibody by either in vitro method or in vivo method in mice. Then we will purify it and then we will store it. So these are the steps of hybridoma technology. Now based on the type of antibody, uh, whether the origin is from mice or human, uh, the name will change. So usually the name of monoclonal antibody will end with the MAB, that is monoclonal antibody, MAB. So if the origin of this monoclonal antibody from murine, which means from the rat, usually the monoclonal antibody name will end with OMAB or MAB. And if the monoclonal antibody origin is from human, which is chimeric, almost 65% is from human, usually the name will end with a SIMAB, SIMAB. And if it is more than 90% of human origin in the monoclonal antibody, it is usually known as SUMAB, and 100% human will become UMAB, UMAB. So what is the importance of this percentage of fraction which is acquired from the human or animal? As the human fraction increases in the antibody, monoclonal antibody, its immune rejection will be very low. The potentiality of immunogenicity will be very, very low. പുറത്തുനിന്നുള്ള ഒരു ആൻറ്റിജൻ ആയിട്ടാണ് അത് കൺസിഡർ ചെയ്യുക. ഇത് ഈവൻ ദോ ഇറ്റ് ഈസ് എൻ മോണോക്ലോണൽ ആൻറ്റിബോഡി, ഇറ്റ് ഈസ് ഫ്രം സം അതർ സോഴ്സ് അല്ലെങ്കിൽ അതർ സ്പീഷീസ് എന്ന് വരുന്നതാണ്. സോ the immunogenicity will be very, very high in case of murine origin monoclonal antibody. If it is human origin, its immunogenicity will be very, very low. So the human rejection will be very, very low. So in case of this monoclonal antibody, it will be very, very high also. Okay. Now these are the some of the approved therapeutic antibody which is available in the markets. And there are some applications. So this is usually seen as a 2-mark question, applications of monoclonal antibody. So which is mainly used for the diagnostic applications like biochemical analysis, diagnostic imaging, and therapeutic application like direct usage of monoclonal antibody as a therapeutic agent, and monoclonal antibody as a targeting agent. And it is used for the protein purification as well as miscellaneous application. So the first one is diagnostic application. So it is mainly for biochemical analysis like radio immunoassay as well as ELISA, enzyme linked immunosorbent assay. So this can be used for the detection of pregnancy by detecting the urine level of human chorionic gonadotropin, and also for a diagnosis of different cancer by estimation of plasma carcinoembryonic antigen in the colorectal cancer, some hormonal disorder by analyzing the thyroxine as well as triiodothyronine as well as thyroid stimulating hormone by thyroid disorders, and infectious diseases by detecting circulatory level of antigen specific to the infectious agent. So these are the some of the applications of biochemical analysis for the diagnostic application. And second one is diagnostic imaging. The difference is one is for detecting, second one is producing some kind of images. So by using radio labeled monoclonal antibody, which is mainly used for the detection of myocardial infraction. സോ ഇത് നമ്മൾ മയോകാർഡിയൽ ഇൻഫ്രാക്ഷൻ ഉണ്ടോ എന്ന് ഡിറ്റക്ട് ചെയ്യല്ല ചെയ്യുന്നത്, വി ആർ പ്രൊഡ്യൂസിങ് സം കൈൻഡ് ഓഫ് ഇമേജിങ്. എവിടെയാണ് ശരിക്കായിട്ടും ഈ ഒരു പ്രോബ്ലം മയോകാർഡിയൽ ഇൻഫ്രാക്ഷൻ വന്നിട്ടുണ്ട് എന്നുള്ളതാണ്. യൂഷ്വലി can be done after 20 to 48 hours of IV administration of attimyosin monoclonal antibody labeled with radioisotope indium chloride, which is mainly used for the detecting myosin. മസിൽ സെല്ലിൻ്റെ റപ്ചർ ഉണ്ടാകുന്ന സമയത്ത് റിലീസ് ചെയ്യുന്ന മയോസിനെ ഡിറ്റക്ട് ചെയ്യാൻ വേണ്ടിയിട്ടാണ് ഈ ഒരു മെത്തേഡ് യൂസ് ചെയ്യുന്നത്. And also it is used for the diagnostic imaging of deep vein thrombosis after the 4 hours injection of radiolabeled monoclonal antibody against the fibrin or platin can be used. പിന്നെ അതിറോസ്ക്ലിറോസിസ് ഏതെങ്കിലും സ്ഥലത്ത് ബ്ലോക്ക് ഉണ്ടെങ്കിലൊക്കെ അതിൻ്റെ ഇമേജസ് ജനറേറ്റ് ചെയ്യാൻ വേണ്ടിയിട്ട് റേഡിയോ ലാബിൾഡ് ആയിട്ടുള്ള ആക്ടിവേറ്റഡ് പ്ലേറ്റ്ലെറ്റ്സ് ക്യാൻ ബി യൂസ്ഡ്. ആൻഡ് ആൾസോ ഫോർ ബാക്ടീരിയൽ ഇൻഫെക്ഷൻ ആസ് വെൽ ആസ് ഹെമറ്റോപോയിറ്റിക് മാലിഗ്നൻസീസ് ഒക്കെ വേണ്ടിയിട്ട് ഡിറ്റക്ട് ചെയ്യാൻ വേണ്ടിയിട്ട് യൂസ് ചെയ്യുന്നു. ഓക്കെ, നൗ ഇമേജിംഗ് ടെക്നിക്സിൽ different cancers can be detected like carcinoembryonic antigen for associated cancer like colon cancer, stomach cancer, pancreatic cancer, and alpha fetoproteins for liver as well as germ cells of testis cancer, human chorionic gonadotropin for choriocancer carcinoma, prostatic acid phosphate for prostate cancer. So these are the some of the radio-labeled monoclonal antibody for detecting, diagnosing, imaging of the different cancers. Now, second application. So, first one is for diagnosis of different diseases. Second one is therapeutic application. We can use for treatment of some kind of diseases like treatment of cancer, transplantation of bone marrow, organ, autoimmune diseases, cardiovascular diseases. So sometime we will use direct use as a monoclonal antibody as a therapeutic agent. Sometime monoclonal antibody will be used as a targeting agent. So here these are some of the examples of application of monoclonal antibody as a direct therapeutic agent in destroying disease causing organism, in treatment of cancers, in immunosuppression of organ transplantation, in treatment of AIDS as well as in treatment of autoimmune diseases. And for targeting agent, as a targeting agent, monoclonal antibody is used as an immunotoxins in drug delivery to target the specific cell, in disease treatment as well as in the dissolution of blood flow. So these are some of the application as a therapeutic application, and sometime we will use it for an protein purification as well as sometime for miscellaneous application like auto antibody fingerprinting as well as catalytic monoclonal antibody, etc. So these are the applications of monoclonal antibody, which is usually seen as a 2-mark question, and these are some of the examples of monoclonal antibody and its use. So there are some antibody which is used as an anti-inflammatory agents like infliximab, adalimumab, etransepap, basilizumab, daclizumab, omalizumab. So you can see the last ending word MAB. If it is the name is ending with MAB, that indicate monoclonal antibody. If it is ending with umab, it indicates it is from human origin. So it is mainly used for different anti-inflammatory conditions like rheumatoid arthritis, Crohn's disease, ulcerative colitis, etc. So these are some of the uses of monoclonal antibody as an anti-inflammatory agents. And then there are some monoclonal antibody which is used as an anti-cancers like gumitizumab, alemtuzumab, rituximab, transituzumab, nimotuzumab, cetuximab, ബാസിസിമാബ് എന്നൊക്കെ പറയുന്ന കുറേ സാധനങ്ങളുണ്ട് ഫോർ ഡിഫറെൻറ്റ് പർപ്പസസ് ലൈക്ക് റിലാപ്സ്ഡ് അക്യൂട്ട് മൈലോയിഡ് ലുക്കീമിയ, B-cell leukemia, non-Hodgkin lymphoma, breast cancer. So these are some of the applications or uses of monoclonal antibody for the treatment of cancer. And there are some other like for the treatment of some kind of viral infections, palivizumab, abcicimab, coagulation. It is mainly used for the prevention of coagulation in coronography, angioplasty, etc. So these are some of the FDA approved therapeutic monoclonal antibody. These are some approved dates. So this is not an newer discovery. Actually this were there on 20-25 years ago. Okay, ഇപ്പോൾ അതിൽ ലേറ്റസ്റ്റ് ആയിട്ടുള്ള ഡിസ്കവറീസുകളൊക്കെ വന്നുകൊണ്ടിരിക്കുകയാണ്. ലാസ്റ്റ് വീക്ക് യു മേ റീഡ് സം ന്യൂസ് റിഗാർഡിങ് there is an discovery in the Russia for vaccine against ക്യാൻസർ ഇൻ ദ റഷ്യ. അത് ഫൈനൽ സ്റ്റേജിലാണെന്നുള്ളൊക്കെ ന്യൂസൊക്കെ നിങ്ങൾ ചിലപ്പോൾ വായിച്ചിട്ടുണ്ടാവും. സോ പ്ലീസ് അപ്ഡേറ്റ് യുവർ നോളജ് വിത്ത് ദ ലേറ്റസ്റ്റ് ഇൻഫർമേഷൻ അബൌട്ട് latest discovery of monoclonal antibodies. So even though there are many applications, there are some side effects by using monoclonal antibody. So there are some more common side effects like allergic reaction, like itching, skin rashes, flu-like symptoms including chill, fatigues, etc. There are some rare and more serious side effects, infusion reactions like severe allergic reactions, dangerously low blood cell count. It may cause decreasing RBC, WBC platelets, and there are some cardiac complications as well as bleeding chances. Even though they are rare, but they are more serious. And there are more common side effects which are not too much serious. So these are some of the side effects or ADR of monoclonal antibody. So these are the important questions from this area. It is usually seen as an essay question, as a short note question, as a 2-mark question, like application of hybridoma technology, explain the method of production of monoclonal antibody by hybridoma technology. So based on the mark, you have to elaborate the steps in detail. Anyhow, you have to write the definition, you have to enlist the steps of monoclonal antibody production steps, and based on the mark, you have to elaborate the steps. So these are some of the references for the production of monoclonal antibodies, and thank you for your patient listening. That's all about the production and applications of monoclonal antibody. Hope it is clear. Thank you.